All right, and welcome back, genetics kids. Today we're going to talk about section 2 of our chapter. So chapter 10, gene action from DNA to protein. We're looking at protein synthesis. And section 2 deals with the translation of a protein. So in part 1, we talked about how we use the DNA to create RNA, specifically mRNA, which is messenger RNA. And then when we took the messenger RNA, we know that we processed it and then we sent it out into the nucleus, out of the nucleus to the cytoplasm to do the next step of protein synthesis. Well, protein synthesis has 3 steps: transcription, which we already talked about, translation, and protein folding. Now, we're not going to talk a whole lot about protein folding. In fact, I kind of added it into this one a little bit. So we're actually going to finish up how to build a protein in this one. Now, that doesn't mean we're done with the chapter. We still have one more section of notes, but we'll at least have an idea of how to build a protein. So let's get going, shall we? So first, we're going to talk about deciphering the genetic code. So that means, how is it that we can read the information in the DNA and the RNA in order to figure out how to properly put a protein together? And part of it is due to what we call the triplet nature of a reading frame. Now, the reading frame is simply the mRNA that you are looking at in order to build or assign the different amino acids to their proper place. So, Crick and his coworkers were the ones who kind of studied this, and they did confirm the triplet nature. And what that means is that there's a very, very particular reading frame for any gene. So it's a particular sequence of amino acids in a protein that is encoded from a certain point in a gene, meaning that you start at one point, one particular base, and you go to a certain other base, and that is what you read in order to figure out the gene. Now, if you add or subtract 1 or 2 DNA bases, it completely disrupts the reading frame, which means that every amino acid after that point is going to be affected by this. If you only change one base, or you subtract 3 amino acids, then you'll have a change in 1 amino acid in the DNA, or you'll miss 1 amino acid in the DNA, but the rest will all be the same. So adding or subtracting will disrupt the reading frame if it is not in multiples of 3. Adding or deleting 3 contiguous bases, meaning ones that are right next to each other, so they are in a row. They are sequential. That's the word I'm thinking for. They're sequential. It'll add or delete add a new amino acid or delete an existing amino acid from the protein, but all the other amino acids should remain the same. Now, if you remember, last time we talked about these things called codons, and that's kind of what's being referred to in, I'm actually going to go back to the previous picture. So codons, here is the mRNA in the bottom, and you see 1, 2, 3 bases together make up 1 codon. So here in this picture, we have 6 bases. That means we have 6 codons. So that does help, because we know that each of these codons is going to code or specify a particular amino acid. So if we were to completely take out this ACA, then that amino acid would no longer be present, but the other codons behind it would be unaffected, which means their amino acids would be unaffected. However, if I take out this A right here, the first A and the second codon, that's going to shift all the other amino acids, bases forward, which is going to affect all of the different amino acids. Now, the genetic code is non-overlapping, meaning that these codons, they don't overlap each other. It's not like if we look here in this picture, AUG, and then the G is part of the next codon, No, each codon is its own separate, unique thing, its own unit. They don't overlap, so AUG isn't going to overlap, meaning be part of any other codon. It's its own particular codon. It is continuous. This process of taking the information, transcribing it, translating it, creating a protein, and then the protein expressing whatever trait it represents is going to continue going on. I mean, we don't have to think about it. We don't have to get it going in motion. It does it automatically. It's virtually universal, which means that we see a lot of similarities, almost identical similarities across all species. Now, there are some slight differences in terms of which amino acids are more common, what are the exact proteins that are being created, but for the most part, we see a huge amount of similarity. The process of translation in me is going to be pretty much the same as a process of translation in a flower or an insect. So it's just kind of cool how that works. We also say that it is degenerate. So what that means is once we're done with the mRNA and we've used it to create a protein, we actually break it down and recycle it. So we can recycle that existing mRNA, break it apart into the separate bases, and send it back to the nucleus where it can be recycled and reused to make a completely new mRNA. Now, Crick hypothesized that there is another type of molecule that needed to be involved in order for translation to effectively occur. And he called this an adapter molecule. And later on, it was actually discovered and we called it transfer tRNA. So it's not so much just that the ribosome latches onto the mRNA and then the amino acids magically line up. There's this other, what he called an adapter molecule that was present, which would bring the amino acids and organize them. And that's what the transfer or tRNA does. I know it stands for transfer RNA. I like to think of it as taxi RNA because it actually brings the amino acids down, puts them in the right place, and then it leaves and go gets another one. But that's an unofficial term. So please, on a test or in conversation with any type of, professors, professionals, colleagues, etc., don't actually call them taxi RNAs. Now, initiation is the first step in translation. So what happens is the mRNA, it's been processed and now it's out of the nucleus and in the cytoplasm. It is going to be involved in this because it has the information. We also have the tRNAs with amino acids bound to one end of them. We also have ribosome, which is the small organelle responsible for making proteins. We have different energy molecules to help or provide energy and, sorry, I was watching my dog stretch, and all that good stuff, plus protein factors, which help to assemble the protein and fold it in the correct shape. So all of these things are involved in this process of translation. We're gonna kind of break it down, as you can see in the picture too, like the very basics and simple aspects, but it's actually pretty intense and pretty crazy how this works. I mean, I know you've heard me say this, I'm a science nerd, especially a bio nerd. This just kind of blows my mind and is really cool that our cells do this all the time. And it's just awesome. Anyways, I digress. So to initiate translation, we start off with the mRNA leader sequence, and it's going to bind itself to the rRNA or the ribosomal RNA in the small subunit of a ribosome. Now remember, the ribosome is kind of made up of two parts. We have the small subunit on the bottom, and then we have the large subunit on top. So the subunit on the bottom, the small subunit, is actually what's going to attach to the leader sequence on the messenger RNA. And then that is going to then allow, allow it to be rooted there so that it can start being read. Don't give me that look, Ripley. Don't give me that look. And then the first codon, so the very, very first codon, which is generally AUG, is going to attract a particular tRNA in order to form what's called the initiation complex. Now, this particular tRNA is very important because that first codon, AUG, in the mRNA, we call it the start codon. And the tRNA that binds to the start codon has a very special amino acid associated with this called methionine. And let's see there. Okay. So once it has there, methionine is on top, it actually sends out this signal for everything else to congregate and get going. So now the large subunit of the ribosome is going to attach to that initiation complex, and it's going to kind of start reading the mRNA in terms of codons. Now the ribosome has slots to hold 2 tRNAs at a time. The first slot is called the P site, and that's the site that is holding onto the growing amino acid chain. The second site is where the next tRNA is going to drop in to add the next amino acid to the growing chain. Now, I do have a video that kind of shows this, how this process works a little bit more smoothly. It's really difficult to talk about and all that, and to visualize at the same time. I'll definitely check out that video less. So as you can see on the picture, here's the mRNA, we have the ribosome, and these yellow squares are the tRNAs. And these little circles, those are the amino acids. So this is showing it midway through. Notice how there are 2 tRNAs inside the ribosome that are connected up with the mRNA. And we said the mRNA has codons, a sequence of 3 bases that will dictate which amino acid should be next. The question should be, how does it know? Well, because the codon in the mRNA actually has a complementary anticodon in the tRNA. So here, if you look, we have AAA as a codon, and the tRNA has a complementary anticodon, UUU. This first one here, UGG, that was its anticodon, ACC. And then GAU, anticodon CUA. You have UUC, anticodon AAG. So that's how they know how to line up, because the codons and the complementary anticodons are going to temporarily bond while they're in the ribosomal complex. So notice this first tRNA here that's in the first spot, the P sites of the ribosome, it's holding the chain. This amino acid here was the first one. This was the one that this tRNA added. In the second slot, the S site, we had another tRNA drop down. Now that it's holding its own amino acid. And once these two are close like this, they can form a peptide bond. So a peptide bond forms between the 2 amino acids. At that point, once this bond forms, this tRNA is going to break its bond with its amino acid, and it's going to leave. The ribosome shifts over. This guy is now in the P site, and this guy can drop into the S site and continue. Now, since we have a chain of amino acids that keeps growing longer and longer, we actually do give this amino acid chain a particular term. We call it a polypeptide, and that's why we call the bonds between them peptide bonds. So we're going to keep building and building this particular polypeptide until we get to the third step of translation, which is termination. So what happens is, eventually, at the end of the mRNA, there's going to be a particular codon that is reached. And the thing with this is the tRNA that binds with it, that has the anticodon, actually doesn't have any amino acid attached to the other side. That's why we call these stop codons. And what happens is when it reaches this last codon, this stop codon, and the tRNA that drops in has no amino acid on the top, it signals that we are done building our polypeptide, our amino acid chain. So once we reach that point, everything stops. And actually, not only will it stop, it will also let go. So the ribosome will let go of the mRNA and the tRNAs. The tRNAs, if there's any left to have, whichever tRNA is in the P site, will let go of the polypeptide chain. The mRNA is free, and we are left with a polypeptide chain, so a chain of amino acids. Now, translation is efficient and economical. What we mean by that is because of the codon-anticodon system, it's very easy to line up the amino acids in the proper order. And it's economical because a lot of these things get reused. The tRNA, the ribosomal RNA, the ribosomes, the enzymes, the other proteins that are associated with this process, they don't even get broken down. They just hang out and wait for the next mRNA to come take its place. So we don't have to rebuild all these things. We don't have to rebuild the ribosome because we don't have to rebuild the rRNA and the proteins that come together to form it. We don't have to rebuild the transfer RNAs. We don't have to rebuild the different enzymes that allow this to run. We don't have to rebuild the chaperone proteins or anything else that helps with this process. It all stays in the cytoplasm. The only thing that we might have to rebuild would be the mRNA, depending on if we need to make more of it, in terms of like a new mRNA we'll have to make to express another gene, or certain amino acids. Now, some of them we can get naturally, some of them we get from our diet. So again, it's all good. All right, so here's a diagram, but kind of all blown up. This is a tRNA that used to be in the P site. So we have the mRNA, the ribosome, these are the tRNAs, and these are the amino acids. This is showing the amino acid chain or the polypeptide chain. So this one used to be connected right here. The ribosome was shifted backwards, and the codon UGG matched up with the anticodon of this tRNA, ACC. He had this particular amino acid bonded to him. So he was in the P site here, holding on to his amino acid and the rest of the chain. This tRNA dropped into the S site, AAA for the codon, UUU for the anticodon, and that leaves us with this particular amino acid. Now, once these two amino acids bonded, they formed that peptide bond. Then the bond between this amino acid and this tRNA broke. This tRNA, no longer attached to an amino acid, left the ribosome and goes out into the cytoplasm to grab another one of these particular amino acids. The ribosome slides over, so this tRNA, which was in the S site, is now located in the P site, holding onto the chain. This tRNA, codon GAU, anticodon CUA, drops into this S site and is bringing this amino acid with it. These two amino acids are now super close, close enough to form a peptide bond. Once this bond forms, the bond between this amino acid and this tRNA is going to break. He will leave to go get another amino acid. The ribosome shifts over. This tRNA is now in the P site, and this one can drop into the S site, and so on and so on and so on until we reach that stop codon, in which case a tRNA with no amino acid will drop in, and that will prevent anything else from being added, signaling Hey, we're done building the polypeptide. It's all good. All right. So step 3 of the of protein synthesis is protein folding. We're not going to get into a whole lot of this. The protein can start folding as early as when translation is continuing on. So you can see the words there. So protein folds as translation proceeds, with enzymes and chaperone proteins assisting the amino acid chain in assuming its final functional three-dimensional shape. So what that means is, if you look at this picture here, now a lot of times fully functional proteins are actually the result of multiple amino acid chains that are put together, which is why you see the red, the white, and the blue. So what happens is you could start seeing it kind of folding up a little bit, and then folding up more, and then folding up all together. So whether that's made out of one really long protein, or you have some of these final steps that then bond together to form a larger protein, it needs to be folded up first. The example I like to give is a piece of thread. If I have a long piece of yarn, let's say, it has no function. It can't do anything. But if I take that yarn and I knit it into a sweater, it now has a function. I didn't have to do anything besides fold it back, fold it again. And I took that long yarn and turned it into something with a purpose. And that's what we're doing with these amino acid chains. We're taking these long chains of amino acids and folding them up into a functional form. Now, what the function of that protein is will depend on how it folds, and how it folds depends on the amino acids, where they're at, how many they are, which particular amino acids are where. All of that is going to determine how it folds up, which means it will determine what the function of it will eventually be. If there's any errors in the protein folding, then that could be the basis of some diseases, particularly things like mad cow disease. That's an infectious protein disease. We'll get to that actually in another chapter. But things like that could be the result of improper folding. There we go. All right. Now the question is, if I give you mRNA, could you tell me the amino acid sequence? The answer should be a resounding yes. Yes, you should. And you can do that by using this particular chart here. Now, listen to the words coming out of my mouth. Always use the mRNA with this chart. Do not use tRNA, unless the chart specifically says tRNA. But the vast majority of them, and the ones that we are going to be using in this class and in most classes, is going to be the mRNA decoding chart. So we know that mRNA depends on codons. So it's these codons that will determine the amino acid sequence. Now, how you read this is you start in the middle of the chart and go out. So let's say I have a codon that is CAC. I know it might be a little hard to read, and it might be backwards, I'm sorry. So that is a codon. What I'm gonna do is follow the mouse. We start in the center of the chart, so CAC. That means that it codes for histidine. If I have, since I mentioned it before, let's look at AUG. Again, starting in the middle of AUG. That's methionine. It is the only one of our codon that codes for methionine. That's our start codon. We also have, let's say, UAA. So again, starting in the middle, UAA. Stop. So that's one of the stop codons. Now you might notice that there are multiple codons for different amino acids. For instance, proline. Proline is, I'm trying to find it, there it is. So CCU codes for protein, CCC codes for protein, CCA codes for protein, CCG codes for protein. Now, the reason why we believe that is for what we call redundancy. Let's say that there is a mutation in DNA, but it's in the third spot of a codon. This way, there might be a change in the DNA sequence, but it won't have any effect on which protein is being made because it's not going to change the amino acids. So it's kind of a redundancy to try and eliminate errors due to mutations. So to lead to more silent mutations than harmful mutations. Now again, at the end of this chapter, I will give a link of sources to different videos that will help you out with this. One of those videos is going to be my science in pajamas, looking at protein synthesis, where we actually map it out on my board. And then we'll also talk about the chart as well. So how to use the chart again. Things you need to know with the chart though. Start with the inside, move out. And always use the mRNA. Don't use the tRNA, you'll get an incorrect number. So please, 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 please don't do that. Why don't we do a couple more of these just to make sure that we're understanding it. Yeah, let me just scoot over. All right. So again, we have, let's see, V, A, C. So if you look at the chart, star in the middle, we have G, A, C. That's aspartic acid. But what if I have G, A, G? G, A, G, glutamic acid. So again, we see a little bit of a difference. All right, so I hope that helps. You notice, again, we just have one for methionine, AUG. We do have 3 stop codons though. UAA is stop, UAG is also stop, and UGA is also stop. So we have 3 stop codons, 1 start codon, and everything else codes for amino acids. So hope this helps. Hope that is kind of helps you to understand it and understand the material a little bit better. Again, definitely check out the video. Whether now you can look it up now or wait till the link is posted in the presentation for section 3. I'll probably still post in our Google Classroom though. But if you have any questions, hit me up via Google Classroom or an email. And you guys know I'm here for you. In the meantime, stay healthy and stay safe. All right, you guys, take care. Bye-bye.